Hello everyone, today we are going to see about the agglutination process in the antigen-antibody interactions. So first, antigen-antibody interaction, it is an biomolecular interaction between the antibody and its specific antigen. So these interactions are the non-covalent interactions like hydrogen bonding, ionic bonding, van der Waals force. So these are weak forces, that is the antigen and antibody comes in close proximity and these forces can occur only over a short distance. So in this, we are going to see some terms like affinity. Affinity is the strength which one antigen binding site binds with one epitope. So the force of attraction would be strong only when the shape of the epitope fits perfectly in the antigen binding site. So next one is the avidity. So it is the total strength which one antibody molecules bind to epitope and the involvement of all the antigen binding sites of the antibody. So if we take in immunoglobulin G, it is a monomer and it has 2 antigen binding sites, whereas IgM, so it is a pentamer and it has 10 binding sites. Okay, so as the number of antigen binding sites increases, the strength of overall binding also increases. So next one is the cross reactivity. So it is the ability of an antibody to react with similar epitopes on different antigens. Okay. So next one is the agglutination. So agglutination, it is the clumping of particles together and it is an antigen-antibody reaction that occurs when an antigen is mixed with its corresponding antibody. So the two bacteriologists, Herbert Edward Durham and Max von Gruber, they discovered this agglutination process in 1896. Okay, so the Gruber termed the name called agglutin. So for any substance that caused agglutination of cells, he called that as the agglutin. So next, the French physician Fernand Vidal. So he put Gruber and Durham's discovery to practical use later in 1896, using the reactions as the basis for a test for typhoid fever. So next one is this Vidal's test is the first example of serum diagnosis. Then Austrian physician Karl Landsteiner, so he discovered the ABO blood grouping. So it is also called as the Landsteiner's agglutination test. Okay. So next, this agglutination is classified into two types like active agglutination and passive agglutination. So active agglutination is where the antigens are found naturally on the particles, and for example antigen found in the RBCs and in the bacteria. So in this active agglutination there are different types. Let us see the important tests. Okay, the first is the slide or tile agglutination, then tube agglutination, then Coombs test. First, slide or tile agglutination. So this is a basic type of agglutination reaction performed on the slide. So in this method, a suspension of unknown antigen or a drop of standardized antiserum is placed on the slide. So this is a viral test for the typhoid fever. So the process includes like first we have to collect the blood sample from the patients and we have to allow it to clot. So after the formation of clot, we have to go for the centrifuge. So in that, the serum will be separated from the cells. So then we can take a drop of serum on the slide and we have to add the antigen solution which contains the killed Salmonella typhi and thoroughly combine those antigens. Then we have to observe the agglutination. So if the clumping is there, then it indicates the positive result that the patient has the typhoid fever. So next one is the tube agglutination. So it is also a viral test. So this test is to detect the O and H agglutinins for typhoid and paratyphoid bacilli. So O agglutinin. So it is the antibody against the O antigen, which are usually can be detected 6 to 8 days after the onset of fever. So whereas H agglutinin, it is the antibody against the H antigen. It can be detected 10 to 12 days after the onset of fever. Okay. So at first for O agglutination, they used Felix tubes. It looks like a short rounded bottom tubes And for H agglutination, they use Dreyer's tube. So it is a narrow tube with a conical bottom. But nowadays, 3 into 0.5 ml corn tubes are used for both the O agglutination and for the H agglutination. In this process, we have to take 8 test tubes and label it as 1 to 8. Then add 1.9 ml of saline in the first test tube, and in the rest of the test tubes add 1 ml of saline. Then add 0.1 ml of serum in the first test tube. So we have to achieve the two-fold dilution. So for that, we are taking the 1 ml of the diluted serum from the first test tube and add it in the second test tube. And we have to continue this serial dilution till the 7th test tube. So we can discard the 1 ml of diluted serum from the 7th test tube, or we can use that for the further experiment also. So now the dilution of the serum sample achieved from tube 1 to 7 will be like 1:20, 1:40, 1:80 and so on. Now we have to add the drop of antigen and mix it and cover the test tubes. So incubate at 37 degrees Celsius for overnight. Now the test tube measures the titer of the antibody present in the patient's serum. So for O agglutination, if the titer value is 1:100 or above, the patient is suffering from the typhoid or the paratyphoid fever. So if for H agglutination, the titer value is like 1:200 or above, then it is a positive case. Okay. Next one is the Coombs test. It is also called as the antiglobulin test. So it was discovered by Coombs et al. In 1945. So this is to investigate for the identification of anti-D or RH antibody. There are two types in this Coombs test: direct antiglobulin test and indirect antiglobulin test. So in the direct antiglobulin test, we have to collect the red blood cells from the patients and wash that red blood cell 3 times with the normal saline. Then you have to add the 3% washed cell red cell suspension in the test tube and add 2 drops of Coombs reagent. You have to mix and go for the centrifuge and remix the cells, and you can able to see the agglutination. So this clumping indicates the hemolytic anemia or syphilis or erythroblastis fetalis. Okay. So next one is the indirect antiglobulin test. So this is used to detect the anti-D or the RH negative antibody in the patient's serum to determine if the patient have a reaction to blood transfusion. So in this, the recipient serum is obtained which contains the antibodies. Now the donor's blood sample is added. So the recipient serum targets the donor's RBC to form the antigen antibody complex. Now we have to add the Coombs reagent, and we can able to see the agglutination of the RBC occurs. So next, let us see about the passive agglutination. So it utilizes the particles coated with the soluble antigen or antibody. For example, latex. So there are 2 important tests like latex agglutination and heme agglutination. So first, latex agglutination. So it is carried out by coating either antigen or antibody or an artificial carrier particle called the latex beads. So it is formed from the amorphous polymer, usually the polystyrene. So first, the latex agglutination for the antibody detection. So it is a passive agglutination test with antigen bound to the surface of the latex beads. So the size of the latex beads is 0.81 μm diameter. So in this, the latex particles are coated with the specific antigens. Then for the latex agglutination for the antigen detection, so it is a reverse passive agglutination test with antibody bound to the surface of the latex beads. That is, so the latex beads are coated with the specific antibody which are agglutinated in the presence of the homologous bacteria. So this lattice agglutination test is used to detect the viral infections, autoimmune diseases, and serum proteins. So for both the antigen and antibody detections, common procedures are involved. So it involves the coating microbeads of latex with pathogen-specific antigen or antibody. So the patient's cerebrospinal fluid or serum or urine is mixed with coated latex particles in serial dilutions with normal saline and observed for the agglutination. So application involves to detect the antigen to Cryptococcus neoformans in cerebrospinal fluid or serum. Then to detect the capsular antigen of pneumococcus, Haemophilus influenzae, then meningococcus. And confirm the presence of beta hemolytic streptococcus from the culture plates. So the next one is the heme agglutination. So in this, the certain viruses like hemoagglutinating viruses, they aggregate the RBCs in the suspension. So the heme agglutinating virus, it has an envelope and the glycoprotein is present on that envelope called the heme agglutinin. So the presence of these glycoproteins helps the hemoglobin virus to absorb to the right RBC. So in the absence of virus particles, RBC is precipitated by the bottom of the test tube, giving rise to a red color in the test tube. So the presence of virus particles, RBCs clump together as a result of interaction between the protein in virus particles and RBC leads to the lattice formation. In this case, the RBCs are dispersed as clumps, so red dot is not found. So in this process, we have to add 0.5 ml of normal saline in the test tube and add 0.5 ml of virus in the first test tube. So to achieve the two-fold dilution, we have to transfer the 0.5 ml of diluted solution to the second tube, and from the second tube, we have to add the diluted solution to the third tube, and so on. So we have to add the 1% of washed RBCs to all the tubes and mix well. Then allow the RBCs to settle down for 30 minutes. The highest dilution at which the clumping is observed is the positive cases. So application involves detect and quantify the viral infections such as paramyxovirus and influenza. Then to detect the humoral immune response of the body against any disease or infective agents. Thank you.